Once this thing works, then we can get going. Great. Okay, so in lab today, we are going to be looking at cells undergoing cell division, a form of cell division called mitosis. So we'll learn what mitosis is and how that, what the purpose of it is and what happens to cells during this process. Okay. Our cells, each of our cells are living things. They're either alive or they were once alive. So like any living thing, it goes through a cycle. Let's start over here. A cell is born. It goes through its normal daily activities. Maybe it decides to reproduce. So here's where it's deciding to reproduce, and then it goes through reproduction, and then you can have more cells if that new cell decides to reproduce. It's this continuous life cycle of a cell until that can happen until it dies. So the cell cycle starts when a new cell forms and it ends when the cell divides into two cells. So for a cell to give birth, you start out as a single cell, it grows, and then it splits, and then now you have more cells. Two more. You have two cells when you start with one. We're going to go over these different stages. Because this is anatomy, what I want to focus on is how these cells look, what they appear as under the microscope, and why they appear the way they do. When we look at most of our cells under the microscope, what we saw with the cheek cells, for instance, we saw a cell, a blob of a cell with a nucleus in the center, and that's really all we saw. This cell was likely an interphase. Most cells are in interphase. Represented on this circular part, interphase is this entire part right here. So it's most of a cell's life. During interphase, this is when cells are doing their normal thing, normal development. And this is when they can also prepare for cell division, prepare for mitosis. You can see the outline of the cell here. Here's the plasma membrane. You can see the nucleus here. When you see the nucleus, again, what's inside of the nucleus? DNA. DNA is stored inside of the nucleus. You can't see any individual DNA molecule. It's just a mess of DNA all scattered throughout. So it just looks like a dark shaded area, the nucleus. When we recognize cells in interphase, it just looks like a cell with a dark shaded nucleus. Nothing too special about it. Cells are mostly in interphase if you find one under a slide. When a cell is growing and then it decides to make more cells, since this one cell is going to split into two, what do you need to do to prepare for splitting into two? If one cell is going to become two cells, DNA and what's inside of the cell? Organelles. You need more organelles. You basically need double of everything. So roughly double the amount of organelles, so you just start making enough for two, and you double the amount of DNA. The term for that is replication. So in this last part of interphase, this is when we're making double the DNA and more organelles. I'm not going to test you on these different phases of interphase, so don't worry about those. Just know that interphase looks like this. And this is also, if we're getting ready for mitosis, this is when DNA is replicated, during the last part of interphase. Okay. Once our cell has doubled the DNA and roughly double the organelles, that's when our cell can divide. And that's the mitotic phase, mitosis. What is what is the purpose of mitosis in humans? Can someone give me an example? I have here growth and cell replacement. What's an example of growth? Sorry? Child development, great. When you're an infant, or when you're not an adult, basically, you're continuously growing and developing. We started as single cells, a fertilized egg, and that single cell divided again and again and again until we grew to a baby, and then developed into a teen, and developed into an adult. That process happens because of mitosis. So that's what growth means. Cell replacement. Why do we need to replace our cells?
So, say you get hurt, like you get stabbed, and then you have a hole in your arm.
Say you get a scratch.
Or like a stab.
Sure. Say somehow you're injured and your cells are damaged. Those cells that were damaged are going to die. You want to replace the cells that were damaged. You replace that through mitosis. We talked about how we scrape the inside of our cheeks. The outer layer, those cells are dead. They have to be constantly replaced with new cells that are also dead. But they grow and then they die and then they replace. All the cells on your skin, the cells on the superficial layer of your skin, they're all dead. But you need living cells deeper to them that grow and eventually become those dead ones. Because these cells are constantly getting removed. They're just there as protection. So we need to replace things all the time. So skin is one place where mitosis happens very often. Does anyone know a place where mitosis doesn't happen very often at all? No?
Your brain.
The nervous system, the brain, yeah. Our nervous system generally doesn't undergo mitosis once it's developed. There are some exceptions, some parts of the brain, but it's for one reason why if you have brain damage or spinal cord damage, it's pretty much permanent. There's no way that we know of yet to replace the cells. Cool. Okay, so that's why we undergo mitosis, whether it's regular growth and development or to replace cells that have been damaged. The type of cells that mitosis happens in, the term for these cells are somatic cells. Soma, that prefix soma, just means body. So literally cells of the body. That sounds like it's everything. The only cells that don't undergo mitosis are the cells in your gonads, your testes or ovaries. They undergo a different form of cell division, which we'll get to later. So pretty much every cell in your body has the potential to undergo this process of mitosis. Somatic cells. Okay, we're going to go through mitosis, and you can see that things happen within the cell. We start out with the end of interphase, and then we move into different phases of mitosis. If you read some textbooks, it might say that there are 5 phases. I'll say that there's 4. It's really arbitrary. I'm just going to describe 4 different phases and what happens in each phase. So we'll learn about prophase, metaphase, anaphase, and telophase. Okay, so we've gone from just the blob of a cell with a dark nucleus, that's an interphase. If we get into prophase, the nucleus looks a little bit different. I'm just going to drop bigger so you can see it a little better. When you look at the nucleus, what do you notice about these? What do you notice about what's inside of the nucleus? How do you describe its appearance? So it's jumbled up, but
it's more. I think there's more space in there.
There's more space. It's condensed. Each of these dark things where you can see an individual piece, you can see individual pieces is the difference. Here it's all just a hazy mess. But here, they're lined up. Structures, you see these spindles. So that's a very, hopefully, simple thing to recognize. If you see chromosomes lining up, you must be metaphase.
Are the
Centrioles.
centrioles, yeah, are they actually connected to chromosomes?
Are they actually connected to the chromosomes? Yes, they are. They're extending a special protein molecule and attaching on either side. Yeah. These are called centrioles or centrosomes. I'm not going to test you on those. I want you to be able to identify a cell in metaphase. Okay, we've lined it up. Now we can, what's the next step?
Or the part?
Pull it apart. Those spindles can pull. Great. We'll separate those duplicates. So one copy goes to one side, one copy goes to the other side. You can see that separation here. So it's lighter in the middle because you pulled each copy apart. That's called anaphase. Anaphase. Okay, we've separated things, but we're still a single cell. So what's the last step? Single cell. So what's the last step?
Breaking apart.
Separated into two cells. The last phase of mitosis is called telophase. In this phase, you might have noticed that the nuclear membrane went away. We're reforming a nucleus around each set. So here's a new nucleus. Here's a new nucleus. Those spindles go away. You can see the membrane starting to pinch in. You can see the membrane starting to pinch in here. The actual term, so there's the term that describes that pinching in during telophase. Cytokinesis. Has anyone heard the term kinesthesia? What's it? The end goal, at the end of mitosis, now we have two cells. They should, barring mutations, they should have exactly the same DNA. They're genetically identical to each other. We end up with two, they're called daughter cells. That's what they're called. You start with one parent cell, you end up with two daughter cells. They get pulled apart. Telophase, we reform nuclei, but there's two of them, and cytokinesis happens. The cell splits into two. Now you have two fully functioning cells. They each have their own set of organelles, have their own set of DNA. Questions on yes?
Are the organelles always there, or do they form after cytokinesis?
Are the organelles always there, or do they form after cytokinesis? They're there because of what happened in interphase. So during interphase, you replicate DNA and you make more organelles. So it happens in interphase.
And they just know which part to go to.
You've made enough that there's a bunch on one side, there's a bunch on the other. Once it splits, you've got enough to function. You can always make more, but you've got enough to function. Is the process the same for bacteria? It's actually a lot simpler. Without going off too much on this tangent, bacteria don't have any organelles. They don't have any membranous organelles, so it's actually much simpler, much quicker. So they can replicate really fast. So they have to make more DNA, but they still split in a similar process. It's called binary fission. It's called binary fission. Something different. Okay. Let me show you real quick. There's a microscope. The slide that I have here, it's called fish mitosis. So basically, these are fertilized fish embryos. They're fish embryos, and if you look real close, there's 9 of them. There's a grid of 9. They're each a different developing embryo. And so basically it's been sliced so that you can see a cluster of cells in each one of these 9. I'm going to zoom in on one. So remember, we start with the slide at the very bottom. I'm at the 4x objective, 40x total. I'm centering it so that it's one of those circles is underneath the light, and I can adjust the lighting if I need to. And then I can use the coarse focus until I get something, until I get that into focus. So that is the developing fish embryo. This whole thing is the embryo, it's not a cell. These are all cells, all these little things, and all the dark parts, those are the nuclei, the chromosomes. So we gotta look closer. So this is at 4x, I'm in focus. I switch to 10x objective, 100x total. Here's the fine focus, there it is. All right. I can already identify one that's not an interface. Can you see it? This one here. What's this one in? It's metaphase. You can see the chromosomes lined up. Here's another one in metaphase. Chromosomes are lined up. So metaphase always look for a line. Most of these cells, what are they in? Like this one or this one? Most of them, not prophase. Most of them are in interphase. They're not undergoing mitosis. Most of them are in interphase. If we want to find one in prophase, though, what are we looking for? The what? The nucleus is gone, yes, but the DNA is still there. But how does the DNA look? You can see individual chromosomes, but there's no organization to them. So let me zoom in more on this one. This one looks like it's in prophase. You can see individual parts. If I go in closer, just 400x on this microscope doesn't look great on this camera. So I'll give my best shot. Okay, that works. Here it's way smoother, so I would say this is interphase. Here you can see clusters. I would say that's in prophase. So that's in prophase. So we've done prophase, we've done metaphase. If we're looking for anaphase, what are we looking for? Pulled apart. So this is something you're going to be doing. You're going to be searching around for different types of cells in different stages. So let's see if we can find one. See one, let me know.
Oh, right there.
It's metaphase.
Down, down, and down.
Oh, where?
It was like down.
Down?
Middle, down, to the right. Does that one have 2 cells in it?
This one?
Yeah.
It's a little hard to tell. It might be, but I can't say definitively. This one here? Top left. That one also looks weird. I'm not sure what's going on here. I'm going to try to find one where I can definitively say that it's an interphase, or anaphase, excuse me. It's another metaphase right there.
Professor,
I would just pass one. Oh, here's one, not an anaphase, but you see this? What's that? That's telophase. See how the cytokinesis has happened, but it's still slightly attached, and the chromosomes have separated to either side? That would be telophase. This one's pretty hard to find, but that's telophase. So I haven't found anaphase yet. That one looks like it's in prophase. This one here looks like prophase. This one over here looks like it's in prophase. Oh, here's another. Yeah, I'd say this is telophase also. So they're pretty far apart. So this is telophase.
Okay. Like that. Right there.
Left or
By
right?
the cursor, right there,
Here?
to the left.
Left?
There's 2 cells right there. Up.
Up.
Left.
Left.
That one.
This one?
Yeah.
No, because it's kind of smooth. I want it to be more distinct like that. It's separating. So once again, you can see how much trouble I'm having. It's going to take some patience on your part to find one that matches. Let me go to a different embryo. Here we go. Let's try this one. You have 9 of them to choose from, so if one doesn't work out, move to the next one. More metaphase over here. Wait. Okay. This is just the beginning of anaphase. You can see that the chromosomes are lined up here and here, but there's a little separation between them. So this cell is in the beginning of anaphase. This one too, possibly, kind of separating over here. But it's a little more clear here. You can see that separation in the middle. So one of your tasks in your lab checks, which I'll hand out in a minute, is to find different cells in different stages. Any questions on what you're identifying? How to identify? When fertilization happens, when a sperm fuses with an egg cell, that's what fertilization is, the fusion of egg and sperm, you form a single cell called a zygote. The word zygote means to yoke together. Like if you have a, I don't play like organ show, how you like yoke 2 oxen together with a wooden, yoke means combine, join 2 things. So you're joining sperm and egg, zygote. So that's what this is. A single cell is a zygote, a fertilized egg. That single cell continues to divide by mitosis over and over and over again. The cells that we're looking at, the fish, it's at somewhere in this stage. So they're going to be divided pretty rapidly, which is why we're using them to look at mitosis. At these really, really early stages, all of these cells are basically the same. They're stem cells. They have the potential to become anything. But clearly, every single one of our cells in different places are different. You have skin cells, brain cells, liver cells, they've all differentiated. The beginning of that differentiation happens early in development around here. I'm not going to test you on when it happens, but I want you to know the different layers. They're called different germ layers. There are 3 primary germ layers when these cells start to differentiate into different kinds. They started out all as the same, but then they become different types. Once you see those germ layers, that's when we know we have an embryo. There's different names for these early parts, so I'm not going to go over them. Things like blastula and gastrula. Once you have these divided germ layers, those germ layers specialize and differentiate, and that's what can become full functional human being. So what are the 3 different germ layers? In that image, it's representing a spherical shape. So this is supposed to be 3-dimensional, a 3-dimensional embryo. There are 3 layers, and they're color-coded here. The very outside, and a little bit of this dorsal posterior layer. This is called the ectoderm. ECTO derm. Ectoderm. The ectoderm will become, since it's on the outside, it would make sense that it becomes things like your skin and eyes. But this little part that goes in here, remember our brain and spinal cord is in those dorsal cavities? Ectoderm also differentiates into the nervous system. Brain, spinal cord, and nerves that branch off of that. So the ectoderm becomes your skin and your nervous system.
Is that why it's so hard to make more of the nervous system, because it's the first thing that was made?
Is it hard? Is it because the nervous system is the ectoderm? Is that why it's hard to make more? That's not the major reason why. It has to do with, I don't know. I can't even give a real answer. It has to do with its programming, like just the way that its DNA can be handled and regulated. It has to do with that, and that gets into stuff that I'm not an expert in. So, good question. Okay, so that's the very outside, ectoderm. The very, very inside, this white space, what would you call this white space? Like once it develops into a full human. Any guesses what this space would be? It could be a lumen or a cavity. Spaces within your body, lumen or a cavity. So what's lining this very interior space? That's the endoderm. Remember, endo means inside. Endoderm. The endoderm, since it's the inner lining, it becomes things like your gut, so like your esophagus, stomach, intestines, liver, and your lungs. The endoderm is whatever's lining a space inside of you, lining spaces. Our third layer is in between those two. So it's all this stuff shown here. So it's in between endo and ecto, or endo and ecto. This is the mesoderm, meso meaning middle. So the mesoderm, that will differentiate into things like a lot of different things. Muscles, blood, bones, pretty much everything else. Everything that's in between the skin and your lining minus your nervous system. Everything else. So as far as what I could test you on this, yes, I want you to know what they each differentiate into. But if I were to show you a diagram like this, and I pointed to one of these, you could identify it as endoderm, ectoderm, or mesoderm, and what it could become. So we've gone over mitosis, or really the cell cycle. Interphase is the major part of the cell cycle, so most of the cells you'll see are in interphase. A small part of the cell cycle is mitosis, and that's when division happens. So it doesn't take up that much time, just like having babies doesn't take up that much time. Just a fraction of a cell's life. And it's this process from fertilization to developing a full adult human, that's when mitosis is primarily happening. Somatic cells. We'll talk about that other half later on. Take a moment, please. Raise your hand a few more times. Okay, hands on three. 1, 2, 3. Three, say letter. 1, 2, 3.
B.
B, yeah, skin. Skin is constantly reproducing. There's a really high rate of mitosis. So if you wanted to take a sample from a different part of your body, you're more likely going to see mitosis happening with the skin than the brain, lungs, or muscles. It can happen in these other places, it's just much less frequent, the brain being the least frequent. Raise your hand a few more times. Hands on 3. 1, 2, 3. Hands. Talk to your neighbors real quick. All right. One more time, please. Let's get hands. 1, 2, 3. On 3, say letter. 1, 2, 3.
C.
Yeah, ectoderm. That's what the nervous system stems from. We're talking about the spinal cord. That's part of the nervous system. Ectoderm. Okay. If you want to take a break right now, this is a good time to do it. But what I've handed out to you before you do take a break, what I've handed out to you are 2 different things. One, I've just decided to print out the entirety, not the entirety, but these are study guides for each lab exercise. So here's lab exercise 1, 2, all the way up to 10. We'll get to in the next, I don't know how many weeks, but. So this is just reference. There's nothing to do with this besides use as a reference. So yeah, that's just for your reference. The other thing I've handed out to you is this lab check 2. You just finished lab check 1, so this is lab check 2. You all should have groups of 4-ish. You don't have to stay part A the whole time. You can if you want to, but decide amongst your groups which part you're going to cover, and that will be your part for due next Monday. Same idea, just like we've done before. Don't forget your explanations. Each person, there's stuff on here that we haven't gone over yet, but what we have gone over is mitosis. So I encourage you to get started on the mitosis parts. Don't forget, we have sketch sheets over here. I want all of your sketches to be on these sheets. Use colored pencils. If you need colored pencils, you can get that in this drawer over here. Colored pencils are in this drawer over here. So everything you need is in this area. And around 1:30, we'll start talking about epithelium. So I'll give you time to work and take a break and work. But around 1:30, we'll talk about the next chapter, which will appear on the quiz. All right? Don't forget about when we take microscopes out to check that they're put away properly. And when you put them back, you have to check it with one of us. All the slides that you need are in the back. They should be labeled. So take a look at the back, all the slides of this. Fish mitosis.
Oh, Keith, you got one?
This has to be. And you know that it's not consumable. I always forget. You couldn't forget. Different systems. I'm not going to finish this today. I'm going to go to like. So I'm going to go to. I'm just going to get to talking about. I want to get to talk about the different types of stratified. I just want to talk about these different types of worldviews. So I recognize these characteristics of stratified. So Wednesday I'm going to have this much. I might even skip these other types of strings just to get to this. You contain the basics. Yeah, my quiz contains the basics of the epithelium.
No, no.
Different types of strings. Make sure you when you open up a quiz to change the date because it's from last. Yeah.
On the